Hello, I am Joala Judalu, a VetMed student from the College of Veterinary Medicine through DVMA, and today, together with my groupmates, Regine Grace Cristobal, Clea Bancas, Alexa Francine Pagayon, Angel Gino Blaza, and Marlene Grace Agad, we are going to discuss about noradrenergic or adrenergic transmission, highlighting the synthesis of NOR-A as well as its storage and release, the adrenergic receptors involved, destruction or the dissipation of action of NOR-A, and other peripheral mediators. So, without further ado, let's begin. Before we go deeper into the discussion, let's start with defining what is noradrenergic or adrenergic transmission. According to Prasad and Kohli in 2006, it is a transmission mediated by noradrenaline or norepinephrine at postganglionic sympathetic nerve terminals and that of norepinephrine and dopamine in CNS. In the biosynthesis of NOR-A, tyrosine is converted into norepinephrine by a number of biochemical processes in the sympathetic nervous system's postganglionic neurons and adrenal medulla. Tyrosine is primarily converted to dopamine in the cytoplasm, whereas dopamine is primarily converted to norepinephrine by dopamine beta monoxygenase inside neurotransmitter vesicles. Dopamine, which is produced indirectly from either the non-essential amino acid tyrosine or the essential amino acid phenylalanine, serves as the direct precursor of norepinephrine. Tyrosine is the most prevalent of these amino acids, which are found in almost all proteins and are therefore obtained by eating food that contains proteins. Phenylalanine is converted into tyrosine by the enzyme phenylalanine hydroxylase, with molecular oxygen and tetrahydrobiopterin as cofactors. This process involves the hydroxylation of aromatic ring. Tyrosine is converted into L-DOPA by the enzyme tyrosine hydroxylase, with tetrahydrobiopterin, oxygen, and ferrous iron as cofactors. L-DOPA is converted into dopamine by the enzyme aromatic L-amino acid decarboxylase, or also known as DOPA decarboxylase, with pyridoxal phosphate as a cofactor. Dopamine is then converted into norepinephrine by the enzyme dopamine beta monoxygenase, or formerly known as dopamine beta hydroxylase, with oxygen and ascorbic acid as cofactors. Norepinephrine itself can further be converted into epinephrine by the enzyme PNMT with S-adenosyl L-methionine as cofactor. In adrenal medulla, there are two types of chromaffin granules, one having norepinephrine and the other has epinephrine. Norepinephrine formed in the one type of granules leads into the cytosol, where it is methylated to form epinephrine, which will then enter into other granules. The reaction is catalyzed by PNMT, mainly bound in adrenal medulla and in the CNS, where epinephrine serves transmitter function.
Norepinephrine release is controlled presynaptically by neurotransmitters including noradrenaline itself and neuromodulators such as acetylcholine, dopamine, and etc., whose receptors are located presynaptically at adrenergic nerve terminals. Acetylcholine, norepinephrine, or epinephrine, 5-HT, adenosine, PGE, histamine, enkephalin, dopamine, and ATP inhibit norepinephrine release through muscarinic, alpha-2, 5-HT, sub-1, P-sub-1, EP-sub-1, H-sub-1, D-sub-2, and P-sub-2 receptors respectively, whereas epinephrine and angiotensin-2 activating beta-2 and AT-sub-1 receptors respectively release norepinephrine. Tyramine and amphetamine promote norepinephrine release. Norepinephrine also exerts autoinhibitory feedback mechanism in regulating its release from adrenergic nerve terminals. Normally, in the process of release of the transmitter, when the polarization reaches a nerve terminal, the nerve membrane-bound adenyl cyclase is activated, causing formation of CAMP from the ATP. CAMP in turn helps in opening the Ca channels, resulting in the free influx of calcium ions into the nerve terminal, facilitating fusion of membranes and exocytosis of norepinephrine and ATP from the vesicles. In the autoinhibitory feedback mechanism, the released norepinephrine activates the presynaptic alpha sub-2 receptors, resulting in the inhibition of adenyl cyclase, closing of Ca channels, and the resultant failure of exocytosis.
During the distraction or dissipation, noradrenergic as well as other catecholamines are enzymatically degraded by extracellular monoamide oxidase or MAO and extracellular catechol-O-methyltransferase or COMT. Monoamine oxidase is a mitochondrial surface-bound enzyme present in adrenergic terminals, liver, and intestinal epithelium. COMT is present in many neuronal and non-neuronal tissues. The end product of MAO oxidation of noradrenergic is vanillyl mandelic acid or VMA. Elevated urinary excretion of VMA is used in diagnosing pheochromocytoma in men. The termination of transmitter action of noradrenergic is mainly achieved by reuptake back into the presynaptic adrenergic terminal, from where it is released in uptake 1 and partly through uptake in extraneuronal tissues in uptake 2 and enzymatic breakdown by COMT. The reuptake replenishes the vesicular noradrenergic stores in the nerve terminal. Reserpine makes more of noradrenergic available for breakdown by MAO and ultimately depletes the vesicular stores of the transmitter. Tricyclic antidepressants like disipramine and cocaine or related local anesthetics interfere with noradrenergic uptake 1, causing enhanced peripheral and central sympathetic effects. Most of the released or circulating epinephrine is removed through uptake 2.
Adrenergic receptors are cell surface receptors that become active when they contact a particular class of neurotransmitters known as catecholamines. Catecholamines play a role in sympathetic nervous system's activation of organ, which aids in bringing on the fight or flight response. The alpha 1, alpha 2, beta 1, beta 2, and beta 3 receptors are among them. To produce a variety of physiological effects, adrenergic medications will bind to one or more receptors.
Alpha-1 adrenergic receptors are G-protein-coupled receptors that are involved in neurotransmission and regulate the sympathetic nervous system through binding and activating the neurotransmitter, norepinephrine, and the neurohormone, epinephrine. These receptors are found in smooth muscles, blood vessels, bronchi, GI sphincters, uterus, bladder, sphincter, seminal tract, iris radial muscle, liver and salivary glands. Depending on the location, alpha-1 can produce effects such as constriction, contraction, relaxation, glycogenolysis, and potassium ion release. The two adrenoreceptors play an inhibitory role in the central and peripheral nervous systems by being found on pre- and postsynaptic neurons, members of the 7 transmembrane superfamily of receptors. Alpha-2 adrenoreceptors are triggered by catecholamines, norepinephrine, and epinephrine. Alpha-2A, Alpha-2B, and Alpha-2C are the 3 subtypes of the alpha-2 adrenoreceptor. These receptors are found in the blood vessels, GIT, B islets of pancreas, platelets, brainstem, heart, and salivary gland, which produces various effects depending on its location. Beta-1 adrenergic receptors control heart rate and myocardial contractility. However, under stress and in response to epinephrine release, cardiac beta-2 receptors are also stimulated, leading to further increases in heart rate and contractility. Beta-1 adrenergic receptors are mainly found in the heart and salivary, which they produce certain effects such as increased rate and force of contraction and amylase secretion. Numerous physiological processes in the body, particularly those involving the cardiovascular and pulmonary systems, are thought to be regulated by beta-2 adrenoreceptors. Beta-2 receptors are found in smooth muscles like the blood vessels and bronchi, uterus, bladder detrusor, seminal tract, ciliary muscle, GIT, liver, mast cells, and skeletal muscle. Beta-2 receptors produce effects such as dilation, relaxation, glycogenolysis, and inhibition of histamine release. Lastly, beta-3 adrenergic receptors are found on the cell surface of both white and brown adipocytes and skeletal muscles. They are responsible for lipolysis, thermogenesis, and the relaxation of intestinal smooth muscle.
There are other chemical mediators present in the nervous system, and they are subdivided into non-peptide mediators and peptide mediators. For the peptide mediators, this includes neuropeptide Y present in postganglionic sympathetic nerve terminals. Its effect focuses on facilitation of vasoconstrictor effects of noradrenaline. The vasoactive intestinal peptide, or the VIP, is another mediator which is present in the parasympathetic postganglionic nerve endings. This mediator causes vasodilation in salivary gland, increase in salivary secretion, bronchodilation, and increase in motility of the gastrointestinal tract. GnRH, a mediator present in the enteric sympathetic ganglia, contributes to slow depolarization of postganglionic membrane. The substance P, present in sympathetic ganglia and enteric neurons, contributes to slow depolarization, facilitates in pain sensation, and promotes inflammation. The CGRPB, or the calcitonin, gene-related peptide, has effects similar to substance P. Lastly, for the peptide mediator, is the ovoid peptides beta-endorphin, pulsine-enkephalin, methionine-enkephalin, and dynorphin. These are formed and released from the CNS or from nerves or other cells, and their effects act like opium analgesics.
For the non-peptide mediators, this includes 5-HT present in the intestinal neurons, which produces effects in the GIT, particularly peristalsis. Another type of a non-peptide mediator is histamine, which is present in vascular and non-vascular smooth muscles, which causes relaxation and constriction respectively. It is also present in the gastric parietal cells and sensory nerve endings. Dopamine is also a non-peptide mediator present in adrenergic neurons that causes renal vasodilation and increased cardiac contractility. GABA present in enteric neurons causes inhibitions of peristalsis. The last type of a non-peptide mediator is the NO, present in postganglionic parasympathetic neurons that can cause vasodilation in corpora cavernosa in penis.